Hello everyone, welcome to my channel. In this series, we'll go through previous year IIT JAM questions topic by topic. Today we are starting with a very important topic, structure of bacteria. Now, without wasting much time, let us move on to the questions. First question, in a prokaryotic cell, the presence of electron transport chain is associated with mitochondrial membrane, nucleoid, plasma membrane, or endoplasmic reticulum. So, as we all know, prokaryotic cells do not contain any mitochondrial membrane for electron transport chain. So, the electron transport chain ETC is carried out in the plasma membrane. So, the correct answer is C. Now, next question. Which of the following transport process is not involved in the uptake of nutrients in prokaryotes? Passive diffusion, facilitated diffusion, group translocation, or active transport? So the correct answer to this question is passive diffusion, because prokaryotic cells have rigid cell walls and membranes that do not allow many nutrients to just diffuse in freely. Only very small molecules like oxygen, carbon dioxide, or water can do that. For actual nutrient uptake, they rely more on facilitated diffusion. So the correct answer is passive diffusion. Next question. Peptidoglycan is a heteropolymer of polysaccharide and peptide, polysaccharide and lipid, polysaccharide and nucleic acid, or protein and nucleic acid. So now peptidoglycan or murine is the main structural component of bacterial cell wall, especially in gram-positive bacteria. Now heteropolymer means this heteropolymer word, this means it is made up of two different altering subunits. Those subunits are NAC and NAM. NAC, that is N-acetylglucosamine and N-acetylmuramic acid, NAM. So the correct answer to this question is peptidoglycan is a heteropolymer of polysaccharide and peptide. Peptidoglycan is equals to polysaccharide backbone plus peptide cross links. The polysaccharide part is the altering NAC and NAM sugars, and the peptide part is the short amino acid chains that cross link the polysaccharides. I hope this question is clear. Now, next question. Lipopolysaccharide is present in the cell wall of, very easy question, very basic. Lipopolysaccharide LPS is a unique component found in the outer membrane of gram negative bacteria. Now LPS has majorly three main parts. The first one is lipid A, core polysaccharide, and the third one is the O antigen. Just for knowledge, lipid A anchors the lipopolysaccharide in the membrane, also called endotoxin, responsible for fever, septic shock. Core polysaccharide connects lipid A to the O antigen, and O antigen is highly variable, helps bacteria invade host immune system. Next question. Teichoic acid is found in. Teichoic acid are present in the cell wall of gram-positive bacteria. Gram-positive bacterial cell wall, the correct option is A. The functions of teichoic acid is it provides rigidity to the cell wall, plays a role in cell shape and cell division. So the answer is teichoic acid is present in gram-positive bacterial cell wall. Bacterial ribosomes are. The correct answer is B, that is 70S. The 70S is equals to 50S plus 30S. This 50S is the large subunit and 30S is the small subunit. In eukaryotes, the ribosomes are 80S, in which the 60S is the large subunit and the 40S is the small subunit. Which of the following statements is correct for gram-positive bacteria? First, they possess an outer membrane, they are devoid of peptidoglycan, they possess thick peptidoglycan, they stain pink by gram stain. So which of the following statement is correct for gram positive bacteria? The answer is they possess thick peptidoglycan layer. We studied it in the previous question also that they have a thick layer of peptidoglycan. Next question. The bacterial flagella. The bacterial flagella is first option composed of flagellin covered by plasma membrane, 9 plus 2 microtubule arrangement or composed of cellulose. So bacteria, it is a prokaryote. So the cell flagella is composed of a protein called flagellin. You might be thinking about this 9 plus 2 microtubule arrangement. So let me clear कि 9 plus 2 microtubule arrangement is found in eukaryotes. It is found in eukaryotic flagella, not in prokaryotes. So the correct answer is option A. It is composed of flagellin. The 9 plus 2 microtubule arrangement is found in eukaryotic flagella and cilia. And yes, it is made up of tubulin. But if they are asking for bacteria, this means they are asking you about prokaryotes. So it is composed of flagellin. A option is correct. Which of the following statement is correct for transformation of bacteria? So let us be clear first of all with the word transformation. Transformation, a bacterium takes up the naked DNA fragments directly from the environment. The DNA may come from dead material cells that have lysed, or if the DNA integrates into the host genome, the bacterium gains new traits. Example, antibiotic resistant. You must have heard about the Griffiths experiment, which was conducted, showed the transformation in Streptococcus pneumonia, about the S and the R strain, the virulent and the non-virulent one. So transformation is the uptake of naked DNA from the environment. So option C is correct. They take up naked DNA. Next question. Which of the following is not a method of sterilization? Now, autoclaving. You just what is sterilization first of all? Sterilization is the complete removal or killing of all the forms of microorganism, including spores. Now, if we talk about autoclaving, there all the microorganisms are removed, killed. Autoclaving uses the principle of heat and pressure, where you generally keep the things at 120 degrees Celsius, degree centigrade, and it around at 15 psi for 15 minutes. If you are lowering the temperature, you can increase the time limit. Or so autoclaving is generally going to kill all the microorganisms. Let's talk about freezing. Yes, microorganisms require a proper temperature for their survival, for their growth, and in freezing, if you keep them at a lower temperature, they'll not be able to survive. Radiation may obviously kill the microorganism. Now talking about filtration. Removes microbe by passing a liquid or gas through a filter with tiny pores. It does not kill any microbes. It just physically removes them. Viruses are very small bacteria, can pass through some filters also. So filtration is the correct answer. It is not a method of sterilization. Which of the following statement is correct for photoautotrophs? Photo auto Phototrophs. So organisms that use light as their primary energy source. Now see, photo means they are using light for their energy source. Now with this question we can clear more terms. If I talk about photoautotrophs, I'm writing it as PA. So if we are talking about photoautotrophs, this means they use light energy, use light for energy plus CO2 as their carbon source. Abhi I am talking about photoautotrophs. Now let's move on to photoheterotrophs. I am denoting it as PH. They'll obviously use light. The sun represents light. And for carbon source, they depend on organic compounds for their carbon source. So now here they are asking you phototrophs. Photo words means they derive their energy from light. So the correct option is B. Any other option? Is this a MSQ or MCQ? So this is an MSQ. Don't get confused. D option is also correct because they derive carbon from carbon dioxide. So phototrophs. Option B and option D are the correct answers. Great job, everyone. I hope this session helped you understand the structure of bacteria and how IIT JAM questions are framed from this topic. Keep practicing, stay consistent, and remember every single step you take brings you closer to your dream. Thank you for watching, and I'll see you in the next session soon.